Myself Nitish Kumar Kota. I'm a fourth sem BSc program student. This one is basically we'll be discussing about immunology, the study of immune system, its overview, everything about it. Actually, the syllabus regarding the FIUGP fourth sem syllabus, on the third paper where it is given the topic, the immune system topic. So we'll be discussing that only. So this is the overview, overview of the immune system. Basically what we get in case of immune system, it can be classified into two types, like the innate immunity and the adaptive immunity. In case of adaptive immunity, we'll find the T lymphocytes and the B lymphocytes. And in case of T lymphocytes or T cells or T cell immunity, we will find the suppressor T cells, helper T cells, cytotoxic T cells. In case of B cell immunity, we'll be finding the plasma cells. We'll be finding these B cells, memory B cells. In case of plasma cells, after you'll be knowing later on, like plasma cells, what they will do, they will secrete the antibodies, which will do this complement cascade thing. That will be on the classical pathway. And in case of innate, in case of innate immunity, which we will be learning today itself. So our class for today will be about innate immunity only. So in case of innate immunity, we will be learning about the physical barriers. Those are the first line of defense actually, including the skin, the mucous membranes, the salivas, stomach acids, the flushing actions of the urines and the tears. All of this, what they do is they stop the infection before it enters the body. The second thing we'll be learning about is the blood-borne. So in case of blood-borne, there is two things given: the complement cascade that we have to learn, including the opsonization, including the agglutination, the other things. But the main thing is we'll be learning today is like the innate immunity itself, but not the complement cascade in this session. We'll be learning the complement cascade in the later on section. First, today we will be learning about the physical barriers, about the innate immunity, the basic concepts of the innate immunity, the physical barriers, and the phagocytes, the phagocytic cells actually. So in case of phagocytic cells, there will be neutrophils, macrophages, dendritic cells, basophils, eosinophils, and necessary killer cells. You all know. What they do is they are all phagocytic cells. Either they will engulf the host cell, engulf the infected cell, which are infected by virus, bacteria, and fungi and other things. They will first they will recognize it, then they will kill it. That's the thing. So this is basically about the overview itself. It's shown that the innate, which is also known as natural or native immunity, their response time is within minutes, so they can respond really fast. They involve this, they are having these cells, the phagocytic cells that I've already told you about: neutrophils, macrophages, dendritic cells, eosinophils, basophils. Or basophils, in case of basophils and mast cells, both of them are having the same property itself. And the last and not least is the natural killer cells. And in case of these soluble factors that have been given here, is the cytokines, which will be included in this topic itself, but these things will keep on coming on the later on sections. So even if you're learning the adaptive and the acquired immunity, even if you're learning topics like hypersensitivity and other things, so these things will come. In case of adaptive, which is different from this innate immunity, their response time is they are having a lot longer response time compared to this innate immunity. And cells involved here in this adaptive immunity are the T lymphocytes and the B lymphocytes. Already I have told you before that these T lymphocyte cells, they are having this suppressor T cells, helper T cells, cytotoxic cells. So they also does the same thing. They kill the body cells which are infected, but their pathway, their way of recognizing and killing are different than compared to this innate immunity cells and the phagocytic cells. So we'll be learning differently, not today actually. And here also, in case of this, the soluble factors which are involved are the antibodies actually, which will be secreted by these plasma cells, as we have discussed here before. So this is basically like hemopoietic stem cell. So in case of hemopoietic stem cell, we get two things, like the common myeloid progenitor and the common lymphoid progenitor. So you can see, like, common myeloid progenitor will having this megakaryocyte, erythrocyte, mast cells, myeloblast. So in case of myeloblast, we'll be having this the phagocytic cells that we'll be discussing: basophils, neutrophils, eosinophils, monocytes. Monocytes itself will give us this macrophage, which will be the largest one. And other than that, I will be discussing the common lymphoid progenitor. This natural killer cell today itself, it will come. So they are having the large granules actually. In case of small granules, small lymphocytes, the T lymphocyte and B lymphocytes will come. In case of the plasma cell, will come under the basis of this B lymphocytes itself. So this is basically the innate immune system. So as we know, innate, from the name itself, it's inborn. It is native to our body. It's inborn, and it has immediate response to the particular infection. And apart from that, they are not that much specific compared to that adaptive immunity. Why? Because they are the first line of defense, and they need like time. Actually, they act first, but they need they lack that memory thing. They lack the memory cells activities. They can't memorize the infection, so they will get the infection the first time only, first effect itself. So they are having that non-specificity character. And actually, innate immune system, they doesn't change with the prior exposure. Whatever the prior exposure it is, it will never change. So that's basically the cells and different things that comes up here. Example, you can say the neutrophils and the different type of cells that will found. We already discussed the macrophages, the dendritic cells, all other things. So the first thing that about innate immunity, it includes the physical barriers and the chemical barriers. In case of physical barriers, already told you about the skin, the GIT, respiratory tract, nasopharynx, cilia, eyelashes, body hairs. Saliva, it will come under chemical barriers, stomach acid, chemical barriers, all other things. They all of them serve as the first line of defense. Sorry for this printing mistake. The closed surface of the skin and the mucous membranes, which already forms a physical barrier, like so, that stops the germs from entering. Okay. There will be substances like acids, enzymes, mucus that will prevent the bacteria and viruses from going. As we know, these all things actually help us. Even though if you are going to the nose things, there will be cilia, there will be hairs, which will act like the physical or the first line, act as a first line of defense. So those are the physical barriers. There are also chemical barriers we have talked about here, right? So there are also certain movements already told in the body also stop germs from settling. For example, movement of hair like cilia. There are these movements like cilia, mucillary movements, which will be in the lung itself, the movements of the bowel muscles. These all things, what they do is they stop the germs, and they act as a first line of defense, since it's a physical barrier. So like they are just like the what you can say, the upper guardians, but they are not that much strong actually. They're just like the barrier itself, the simple barrier, I would say. Some fluids in the body have a similar effect, including pure fluid, sweat, and urine. So these are the fluids which flushes the organs of the urinary system. So these are informatives actually. I don't want to go that much, just by reciting. I don't want to go all the paragraphs and all the things that's been written here, but I want to be specific all around what it actually matter. The physical barriers, you have to remember the physical barriers and the chemical barriers, which will act as the first line of defense for the pathogens which will enter the body. So the next thing is the cellular defenses. So in case of cellular defenses, here comes the thing, is like we'll be learning about the phagocytic cells or the immune system cells. And the phagocytic cells actually consist of these macrophages, the neutrophils, which will engulf the pathogens, while natural killer cells, actually, where if you see these phagocytic cells, the list have been given here: the neutrophils, macrophages, dendritic, eosinophils, basophils, mast cells, natural killer cells. So this all among these phagocytic cells, the natural killer cells, they will destroy the infected cell. We will learn here. We'll learn the later. I will learn everything about the natural killer cells today. So the next thing is like the first thing actually, what they do, they circulate throughout the body. The phagocytes circulate throughout the body, and if they see any sort of threats or any sort of like alarm, they what they do is they will engulf it and they will destroy it. They will kill it. That's the thing that will do. The first phagocytic cell that we're gonna learn is the neutrophils. So neutrophils are basically the granulocytes. Granulocytes means they will having this granules here. You see in the diagrams, they are having this small granules. It's a tree-lobe structure. The mid portion of the nucleus is the tree-lobe, but surrounding them are all over this cellular body. Surrounding it is having these granules, small granules. So the granules are there in the cytoplasm. That's why they are termed as the granulocytes. And these are the phagocytic cells. All they do is phagocytosis, and they will engulf the cell. Actually, these granules are toxic to the bacteria and fungi, and they already written, they will stop the cell proliferation and cause the cell death on contact. And the number of these granules are high in the body. Actually, the neutrophils, their number is high in the body, so they act as the first one to serve as at the site of infection. Any infection, any pathogen is coming, it's causing any infection in the body. So these neutrophils will be the one who will be like going to the site of infection, and they will be like the first one which will fight against those bacteria, fungi, and these pathogens. So this is all about neutrophils. I hope everything's clear. So the next thing is the macrophage. You know about macrophage. Macro, like we all know, macro is big, and phage is itself the eater one. So these macrophages are basically phagocytic cells, and what they do is they eat bacteria, viruses, fungi, debris. In case of debris, by eating this debris, engulfing this debris, by killing this debris, what they do, they will be reducing the inflammation and the dead cells itself. So actually, these macrophages, what they do is they use the receptors. We'll be learning all those things in detail in later on, but now it's like the basic things, because we are just discussing about the macrophages. So here bit it's informative itself, but in detail we'll learn it later on. So what they do is they use the receptors. Why receptors should be there in any sort of cell or any sort of body, so that the killer cells, or the the one which will be providing this resistance against these pathogens and all, they will go there, they will kill there. What they will do, they have to bind that, they have to make a contact with that particular host cell or particular infected cell. So what they will do, they will just go for the receptor itself, then only they will get that communication. They can contact with that particular cell, infected cell. So they use the receptor to grab the targets directly, or grab the tag targets. Tag targets means what, like those receptors, what they get is like not so sorry, those target cells, what they having those receptors, what they will get is like they will having these things like complements. So what they do, like if they are having these complements, they will like it's a particularly it's a tag mark, like this is the particular targeted cell, it will be tagged. And already here written, as I've said also, they will be marked by antibodies and the complements like C3b, and there are also C5a, and they will be learning this in hypersensitivity topic. That's a really good topic and important point of view having with this regarding this chapter. So you'll be learning all this later on, but just remember the thing here is like the tag targets, what they will do, they will be marked by these antibodies and the complement proteins. So and also macrophages, what they can do is like they can wrap their membrane around the target cell, and they can swallow into an internal bubble which is known as the phagosome. And they will go there, they will surround the target cell there, and they will swallow it. That's the thing that will do. The macrophages also clean up the body, especially during this apoptosis, and they also clean up the debris there. By cleaning the debris, already I've mentioned that they will reduce the phagos inflammation there. Say they can also act as APCs. Okay, so remember that APCs. The APCs are the antigen presenting cells. So in case of antigen presenting cells, what is there? Like they can process the antigen, whatever the antigen is there. If there's a protein, the protein is coming on, they will process it with the help of this protease or proteasome enzymes or something like that. They will like cut it off, they will process it off, and they will make them minute, like peptides and all. So those peptides, all rest other things, they will be considered as antigens. So these antigens will go there, and antigens will stick with this MHC class 2 molecules. MHC class 2 molecules means the it's a what you call surface protein. MHCs and the major histocompatibility complex are the surface proteins. The surface proteins in the cells actually. What they will do, they will go there, they will form a complex with these MHC class 2 molecules, and they will present it to the helper T cells. They will get out of the plasma membrane, and then actually not out of the, you can say out of the plasma membrane, but they will go there and they will present it to the helper T cells. And what type of helper T cells they will present? Since it's a MHC class 2 molecule, so they will be presenting CD4+. So if it was MHC class 1 molecules, it would be presenting CD8+, but since it's a MHC class 2 molecules, they will be presenting CD4+, helper T cells. And overall, they will alert the immune system. That's how they work. And if the T cell recognizes it as a foreign, they will kill it. They will do whatever it's necessary to do for the T cells. You know the job. And if it's not, then if it's a self cell, if they recognize between this self and non-self. If they recognize it as self one, then they will get continuation will go on, like they will keep on going in roaming around the body. And if it's a like a non-self or the foreign particle, what they will do, they will like kill it and gonna be destroyed. So that's the thing. So apart from that, the macrophages, what they do, they release cytokines. Cytokines are the things which will activate the B lymphocytes, and that B lymphocytes will activate the plasma cells, and the plasma cells activates the antibody or produces the antibodies. Not activates, actually produces the antibodies. That's how it works. That's a basic one. So what they do, they releases the cytokines. So sorry for this, it's written cytokinesis, actually it's cytokines like TNF, IL, interleukin, etc. which will increase the immune cells and causes fever or inflammation. When this will happen, and the antibodies will go there, so there will be fever, there will be inflammation. Later on, they make the anti-inflammatory signals like interleukin 10, TGB to stop this inflammation, and after that, they helps in tissue repairing. That's a repairing thing that goes on. Basically, you have to remember they can also release cytokines, which will increase the immune cells and causes fever and inflammation. That's it. So this is the thing about the macrophages, how it works. You see the what you call the pathogens here. They will go there, and the cell itself, the macrophage itself, they will go there. They like pseudopodia. They will like move their cytoplasm towards this one pathogens. They will engulf it little by little. At the last, they will engulf it and they will destroy it. That's how the macrophages thing that actually works. So the next thing is the dendritic cells. So this is also a phagocytic cell, but apart from that, they are antigen presenting cell. Like if you say isolate in an isolated manner, it's actually antigen presenting cells. What they do is they act as messengers. They act as messengers for the rest of the immune system just by presenting the antigen. What they will just present the antigen in the surface of the particular antigen presenting cell, and they act as a messenger. Actually, their main thing is like this only. You see the diagram. This is the immature dendritic cell. Like inside one is the nucleus, the outside one is the membrane extensions. So when there will be antigen and the T cell will come, along with it, the T receptor will be T cell receptor will be there. So if they are making contact with this, if they are presenting, so what will happen? The membrane extension will get bigger and it will become a mature dendritic cell. So that's how they're gonna phagocyte the pathogens, right, that present here. And basically, apart from that, it is also written that they are basically present in the tissues. So they are the first to know the threat of infection. The tissues will be the first one to get infected. So they'll be the first one to know about the threat of infection. And they can also act like a bridge between the innate and adaptive immune system. Getting my point? They will act as a bridge between innate and adaptive immune system. So this is all about dendritic cells. The next one is the eosinophils. So basically, these are granulocytes only, multicellular parasites. Target this particular granulocytes. Actually, what they do, they target the multicellular parasites. And eosinophils secretes a highly toxic proteins and free radicals that kill bacteria and parasites. Okay, this is the same thing that happens in everything. When there will be granules, what they will do, their cytolytic and the cytotoxic duty will increase, and they will go there, they will kill the bacteria and parasite. They use this toxic proteins and free radicals also cause tissue damage during allergic reactions. So activation and toxin released by eosinophils highly regulated to prevent any unnecessary tissue damage. So this is the thing. So while eosinophils only make 1 to 6% of WBC, and they are also found in many locations, including this is the location of the this eosinophils that's been given there. So this is the particular diagram. You see the granules there, the secretory granules. Those granules will actually like secrete these highly toxic proteins and free radicals, which actually also causes the tissue damage during allergic reaction. So they have to be regulated so that any unnecessary prevention, unnecessary tissue damage happens. They need to take in mind that there shouldn't be any unnecessary tissue damage. So that's the thing about eosinophils. So this one is the mast cell actually. You see the diagrams. So They are found in the mucous membranes and the connective tissues. So it's written there, and important for wound healing and defense against pathogens via an inflammatory response. So when the mast cells are activated, they release cytokines and granules that contain chemical molecules to create an inflammatory cascade. So when the mast cells are activated, they release cytokines and granules that contain chemical molecules to create an inflammatory cascade. So the thing is, when they are not active, so they'll be like there only in the body. But when they are activated, they will release cytokines. Cytokines, what they do? Already told you, they will activate the B lymphocytes, which will actually activate the plasma cells, and that will produce the antibodies. So their main thing is like that only, and they release the cytokines, the granules that contain the chemical molecules to create an inflammatory cascade. Inflammatory cascade is like an inflammatory, what you can say, the reactions, cascade reactions that will go one by one and one and one. So the next thing is the mediators such as the histamine, which will cause the blood vessels to dilate. It increases the blood flow and cell trafficking to the area of infection. So mediators such as histamine and other things, they dilate the blood vessels, which will increase the blood flow and the cell trafficking to the area of the infection. The cytokines, which are released during the process, act as a messenger service, alerting other immune cells like neutrophils and macrophages to make their way to the area of infection or to be on alert for circulating threats. So these cytokines, what they do is like they act as a messenger only. Yes, this is the same thing, which will alert the other immune cells like neutrophils and macrophages, and make their way to the area of infection or to be on alert for circulating threats. So these things that happens. Next thing is the basophils. Basophils actually are the granulocytes. Basophils are the granulocytes that attack the multicellular parasites. You see the basophil, the structure of basophil, it's too low, right? In case of the cytoplasm, the granules are there. They are also granulocytes. So what they do, they will release the same thing, the histamine. That's why I've written the basophils and the mast cells along with it. They will release the histamine, and the use of histamine makes the basophils and the mast cell creep-less in mounting an allergic response. So what they will do, they will fight against the pathogens, and they are actually helping in this, both of them, the mast cell and the basophils, help in this wound healing. They are the key players in case of allergic response. So that's their job. This is about the mast cell and the basophils. So the last one that it came here is the natural killer cells actually, which is also shortly it is called as NK cells. What they do, they are like the 5 to 20%. They are in a great proportion in case of the lymphoid cells, innate lymphoid cells that we have seen earlier. Actually, their name itself depicts that their ability to recognize and kill the tumor cells, the infected cells, virus infected cells. Those are physiologically stressed cells. So to kill these cells which are infected, which are stressed, physiologically stressed, infected, cancerous cells, and to recognize these cells and to kill these cells without presence or absence of any sort of typical markers like the antibodies or the complements for immune system activation. Already written antibodies are MHCs on cells. So this particular set of cells, particular cells are regarded as the natural killer cells. So they don't need any sort of MHCs or antibodies on the cell surfaces for any for their action. They will go there. And they will having the receptors in their body, and if they found those receptor which their activity might get inhibited, or if they're not, then in case of that they will kill that particular target cell, destroy it, blah blah. So the primary site, their location is on of this NK cell development and maturation are the bone marrow. So we'll get everything by the bone marrow itself, and secondary lymphoid tissues, given it's, you can see there. And apart from that, the differentiation and development of these NK cells from the lymphoid progenitor to the medulla NK cells with specific receptors and distinct effector functions rely on multiple intrinsic factors, the transcription factors actually. And apart from that, the extrinsic like the cytokines and the growth factors. There are different cytokines. I have not written every name, but some are common, like interleukins, the anthills, etc. So next topic we'll move forward is the role of NK cells in the innate immune system. So the first thing is like the immunological self-recognition of NK cells. So here basically we'll be discussing about the action of NK cell on the healthy cell or on a normal cell. So we'll be discussing about that thing. So what basically here happens is like the NK cell will be having this inhibitory receptor on their surface. You see the inhibitory receptors. There will be also a activating receptor. In case of healthy cell, there will be the MHC class 1, you see, MHC class 1, and the activating ligand. So here it is written, the NK cells can distinguish between normal cells with stress cells through their surface molecules. Through their surface molecules, they can distinguish a normal cell with a stress cell. And normal healthy cells express MHC class 1 molecules, which are ligands for inhibitory NK cells receptor on the surface. Okay, the MHC class 1 cells, which are actually present in the normal but not in the stressed one, what they do is like they act as the inhibitory NK cells receptors. They will act as inhibitory NK cells receptors on the surface. Basically they will be like the ligands for this inhibitory NK cell receptors, which will be presenting on the surface of the NK cells. And this activation of these inhibitory NK cell receptors in response to the surface MHC class 1 leads to the inhibition of NK cell activation. If the MHC class 1 is present there on the healthy cell, if it's a healthy cell, MHC class 1 will be present there, and in case of that, what will happen? It will act like a ligand to the inhibitory NK cell receptor of the NK cell. And in that case, what will happen? It will lead to the inhibition of NK cell activation. It will stop the NK cell activation procedure. This is about the immunological self-recognition. Self word is already actually used with the good things like the normal cells, the self cell, or the own body cells. So this is the thing. The next thing is about the missing self-recognition of NK cells. The word missing self here has a link, I will say later on. So the first thing is the stress cells. So in case of stress cells, you can say the tumor cells, the cancer cells. Tumor means cancer, the next is so virus infected cells, so fungi infected cells, different sort of things. They transform and they lose their MHC class 1 molecules. So they are deficient. They transform, they modify themselves. So at the time, what happened? NK cells will come. They will detect these abnormal cells. They will detect these tumor cells, virus infected cell. They will not find like the MHC class 1 molecules will not be there in the stress cells. in that cells. And what they will do, NK cell will make it target. So this is the thing we have to act upon. This is the cell that we have to destroy, act upon. And it will trigger the cell mediated cytolytic cytotoxic activity of them through the release of cytotoxic granules. You see the granules here, the red color one, lysis written here, the lytic granules already mentioned. It will consume the granzymes. Granules actually consume the granzymes. They are having the enzymatic activity, including this perforin. Perforin, what they will do, they will like pinch the target cell and they will get entered into it. They will be having these seizure molecules. They will be having these seizure molecules inside them, and what they will do, they will get into the cytoplasm of this infected cell. They will induce apoptosis, programmed cell death, or the cell lysis. So, and in addition to this, NK cells also secrete various antimicrobial molecules and alpha defensins, which will cause cell wall disruption and directly kill the cells. I guess you have understood this line. And simultaneously, the lack of inhibitory receptor ligands. So there will be a lack of inhibitory receptor ligands, which will result in lower inhibition of NK cells and induce NK cell activation. So if the inhibitory receptor ligands will be there and they will result in lower inhibition of NK cells, what they will do, they will induce the NK cell activation. Since there's no inhibitory ligands, receptor ligands are there, there will be no inhibitory ligands. Why there will be no inhibitory ligands? Because the MHC molecules is not there, not present in the stress cell. They are transformed and they are modified. They lack the MSC class 1 and the molecules. So the inhibitory receptor ligands will lack. At this point, what will lack? The inhibition of NK cells will lack, and that will induce the NK cell activation. This is the thing. So it's mentioned as missing cell. Missing cell means missing of this MHC class 1 molecules. So missing cell of the particular this NK cells recognition, missing self recognition of NK cells. So this is the second type, where you will be missing the MSC class 1 deficient. MSC molecules will be not present in the target cell, and those cells will be stressed. In case of the stress one, this particular thing is in case of the stress cells. The earlier one was about the this normal cell, and this one is about the stress cell. The next one is induced self NK cell activation. This one is actually for this stress cells also only, but the thing is it is overly expressed, and since it's overly expressed, what they will do, they will release various cytokines that are ligands for the activating NK cell receptors. They will release the particular cytokines, various cytokines, and those cytokines are interleukin 12, 2, 15, 18, CCL5, all other things. So these cytokines will start circulating. There will be increment in this thing within the affected site, which will increase the expression of activating NK cell receptor. It will increase the expression of activating NK cell receptors, leading to the secretion of key pro-inflammatory cytokines like interferon gamma and TNF alpha. So getting my point, stress cell, when they are overexpressed, they will release various cytokines like the interleukin, various interleukins. and cytokines will act as the ligands for activating NK cell receptors. These circulating cytokines, the increased circulating cytokines will affect the within the affected site. What they will do? They will increase the expression of this activating NK cell receptors, leading to the secretion of pro-inflammatory cytokines like interferon gamma and TNF alpha to activate. And what they will do? This interferon gamma and TNF alpha, what they will do? They will activate various signaling pathways to generate various immune response and promote cell death, apoptosis. So this will happen. And the last line is, with the expression of activating receptors exceeding the loss of inhibitory signaling, NK cell mediated cytolytic or cytotoxicity activity or cytotoxic activity, that's a printing mistake, and secretion of pro-inflammatory cytokines to eliminate target cells are activated without harming healthy cells. Getting what I'm saying? With the expression of activating receptors exceeding, getting more and more than the loss of inhibitory signaling, NK cell mediated cytolytic or cytotoxic activity and secretion of pro-inflammatory cytokines such as the interferon gamma and the TNF alpha to eliminate target cells are activated without harming healthy cells. One more time. With the expression of activating receptors exceeding the loss of inhibitory signals, NK cell mediated cytolytic or cytotoxicity activity and secretion of pro-inflammatory cytokines to eliminate target cells are activated without harming the healthy cells. So healthy cells will not get affected. Healthy cells will not get affected. What they will do? What they will do? If there is a loss of this inhibitory signal, activation will be more. So what they will do? The NK cell will keep on killing, and anti-cell will mediate the cytolytic and cytotoxic activity. They will secrete this pro-inflammatory cytokines to eliminate their target cells, eliminate the target cells. It will get activated. Their process will get activated. So this NK cells main thing is all about this thing only. Like if there is you've seen the second thing, if there is a MHC class protein is there or not on the cell surface. If not, the inhibitory action will get lower. Since the inhibitory action will get lower, the NK cell activation thing will get induced, and what they will do? They will do the apoptosis thing, the cell lysis thing. They will kill the cell, they will destroy the cell. This all happen here also. That's basically what is being given here. So that's all. Thank you so much. And like already I have mentioned here the other things, you can go through it. This roles, the natural killer cells, the basics one, mast cells, basophils, eosinophils. So these dendritic cells and macrophages, they will do the phagocytosis and all. The neutrophils, the cellular defenses, all of these cells that you've seen here, the phagocytic cells, all of them come under the cellular defenses, the immune system cells. They are basically, they are phagocytic cells. We've seen also the physical barriers and the chemical barriers here, and all these physical barriers, chemical barriers, the phagocytic cells, the immune system cells, all of them, they come under innate immune system. And we've already discussed about this thing. This is the chart about the hematopoietic stem cell. This research basically classifies that which falls where and all. This is also another chart about the immunity system, the overview. This is the whole overview about the immune system itself. We'll be learning one by one each and every topic. For today, we have done the innate thing, One thing is remaining, the complement cascade. In the next session we will be discussing about the complement cascade, hopefully. So this is all about, and I would like to thank each and everyone who is eagerly listening to my voice. Since I'm new to this, so I'll not be able to make my communication and make my connection with you guys properly, but promising you guys that I'll be more efficient and more better with my pronunciation, with my vocabs, with the way I'm gonna teach you, and there's more to come. This is the beginning itself. So stay tuned. Thank you so much. Good night.